All right. Well, hello. Good afternoon, everyone. Welcome to the Lunchtime Lecture series from Britt and the Fort Worth Botanic Garden. I'm Dr. Brooke Best, Director of Research Programs, and I and my research colleagues here host this lecture series, which takes place the first Tuesday of every month. So our next talk will be on Tuesday, June 1st, when two of my Brit colleagues, Ashley Bordelon and Harold Keller, will be presenting new fungi and myxomycetes. Let me pop this up on the screen. New fungi and myxomycetes in urban environments, use of live tree bark moist chamber cultures. So they'll be telling you about all kinds of cool local discoveries they've made, including some new species, which is pretty neat. So we are streaming live to YouTube right now. Hopefully that went through. And you live viewers, you can type your questions there in the chat anytime, and we'll answer them at the end of the lecture during the Q&A. When your questions will be displayed on screen, so please try to be brief and ask just a single question at a time. That makes it easier. And if you'd like to test out the chat, please feel free to share where in the world you're watching from today. I want to mention that the lecture and the accompanying live chat are being recorded and will be available in the future on Brit's YouTube channel, pending, of course, permission from the speaker and their home organization. If needed, we will link to other resources mentioned during the talk, either in the chat, and we can also post those to the Brit webpage for this event after the fact. If something does happen and we get cut off or there's a technical difficulty, just please try to refresh the YouTube page or check the event page at brit.org because we'll be posting new information there about possibly reconnecting or switching to a new over to a new link. So that said, let's get started. It's my pleasure today to introduce our speaker, Dr. Rebecca Koll. Dr. Koll is a Paleozoic paleobiologist and a National Science Foundation postdoctoral fellow at Perot Museum of Nature and Science in Dallas, Texas. Today, I believe Dr. Koll will be talking about the Texas landscape of yesteryear. So Dr. Koll, you have the floor.
Great. Well, hi, everybody, and thank you so much for joining me, and thank you, Britt, for having me. As Dr. Bass said, I am a postdoc with the Perot Museum of Nature and Science, and I work on the Paleozoic of what's called Euramerica, or what used to, or many, many years ago, was a supercontinent or part of a supercontinent that made up what's now North America and Europe, and a few other countries are kind of crammed in there as well because, again, it's a supercontinent. So we're looking at all sorts of stuff. I'm actually really excited to be working with Britt and having this opportunity to come and kind of think about living plants because I don't get to do that a lot. But my background is actually in environmental horticulture and crop genetics. And so I spent a number of years working in the public gardens and then also in plant crop research for a little while. So it's kind of a nice break to go back to my roots and be thinking a little bit more about what's going on and kind of, with these experts that are working in the actual modern plants that we're looking at and that we're experiencing in our everyday lives. But like Dr. Bass said, I'm going to be talking today about a look at a seasonally dry ecosystem from the Permian of equatorial Pangaea. So let me just get the full talk up, and here we go. So working in the Paleozoic is especially interesting because the major events that occurred during this time really dictated the future course of ecological and evolutionary history in the terrestrial biosphere. So even though we're dealing with very different species during this time, by the late Permian, the ecosystem structure and dynamics really aren't substantially that different than what we see in even present day. And so the late Paleozoic was this time during which these major radiations were occurring in the plant lineages that came to dominate both the late Paleozoic and the early Mesozoic, but in some cases extending all the way into present. And it's these phylogenetic relationships of key Paleozoic plant groups that can really serve as the framework for helping us understand shifts in diversity and distribution within these different plant communities in response to things like changes in climate, but also in response to disturbance events. So things like fire or insect herbivory or other kinds of big shifts that you're seeing both on the local, regional, and global landscapes. And it's by exploring these rates of species turnover and patterns of species association relative to environmental conditions that can really provide the foundational information for determining patterns of spatial and temporal distribution and informing these kind of bigger biogeographical questions concerning things like the radiation patterns of different plant groups through time, and then also the drivers of terrestrial plant innovation. So things like how plants are evolving, where they're evolving, and if they're evolving or if they're going extinct or if they're just kind of persisting for periods of time. All of that's really important for us to understand. And in short, this is kind of a very long-winded way for me to say that understanding the structure of deep time landscapes and their response to ecosystem drivers really helps us better understand modern ecosystem behavior. So there's a lot we can learn in kind of our modern understanding and kind of extrapolate out to what's happening today and help us predict what might happen tomorrow by looking way back in these deep time environments. And so I'm going to be focusing largely on the Permian of what's now modern north central Texas. But I first want to take a minute to just take a kind of drop the Paleozoic into the broader geologic timescale. So I know a lot of people maybe just think of, you know, honestly, once you get back past modern, it's kind of all just old. So I want to give us a chance to kind of understand where the Paleozoic falls kind of in the broader picture of, you know, life on Earth over time. So the Paleozoic precedes the Mesozoic and the Cenozoic. And a lot of you are probably familiar with the event called the End Permian mass extinction. So this was the massive extinction that created a huge reorganization of both marine and terrestrial flora and fauna. It was at the end of the Permian and going into the Triassic. So it's the big event that kind of separates the Paleozoic and the Mesozoic timeframes. And during this time, during this talk, I'm going to be talking about probably a lot of plants that you might not have heard of. And that's one of the really cool things about the Paleozoic is everything is super weird and that makes it really fun for us to understand. And just to give you some context, the flowering plants, so all of the angiosperms that you might have in your garden. So if you've got your tomatoes and your peppers and things starting to come up, if you're in Texas, or if you're elsewhere in the world, maybe something else is coming up in your gardens. Those didn't evolve for about 100 million years after the Permian. So actually even a little bit longer than that. So we're way, way, way before that. And we're operating in a very different floristic system. And we're also operating in a very different climatic system. So some of the events you might be familiar with are the Paleocene-Eocene thermal maximum. So it's this time period of very extended and heightened warming, or the last glacial maximum, which took place in the Pleistocene. And all of those events are a little bit more similar and probably a little bit more familiar to what's going on kind of in our modern climatic structure. So I'll take you through kind of what's going on in the Paleozoic, but suffice it to say, like the plants, a lot of weird stuff was happening. So, paleogeographically, the Paleozoic is bookended by supercontinents. So you might be familiar with Pangaea, which is the supercontinent that everyone kind of learns about during maybe high school or middle school when you're kind of learning about the history of Earth. But what a lot of people don't know is that there was another supercontinent called Rodinia, and that was before the Paleozoic. So this pre-Paleozoic Rodinian supercontinent really marks the beginning, kind of earliest part of the Paleozoic, because what was happening was this supercontinent was breaking apart. And so as we move through the Cambrian and Ordovician and Silurian into this kind of middle part of the Paleozoic, a lot of what's going on climatically and then also dictating some of the kind of ecological factors with the flora and fauna are controlled by this kind of separation of Rodinia. And then as we move into kind of the later Paleozoic, so into the Carboniferous and the Permian and even into the early Mesozoic, those continent land masses kind of move back together and form Pangaea. And so I know that's a lot, so just to help orient yourself, I've marked with a little star here on the map kind of the general location of where modern Texas would have been. So you'll probably notice first that it's south of the equator. You'll also probably notice that it as well as a lot of North America and a lot of other land masses were under shallow seas. So this actually isn't the worst thing during this time frame, because really all the action was taking place in the marine environments. So the shallow seas and the deep seas were hugely diverse in flora and in fauna. And so it was not a bad place to be. There was a lot of diversification going on. There was a lot of different trophic levels and interactions that are happening. So it's quite interesting. And it's really not until the Silurian, so almost the kind of early to middle part of the Paleozoic, that we start to have the earliest land plants colonizing the terrestrial ecosystem. And as these plants start to kind of radiate and expand in their different life forms and habits, we start to kind of see this expanse and acceleration of landscape shaping processes. And by that, I mean we see kind of an acceleration of weathering processes, which ultimately kind of accelerates and expands the different kinds of soil development that's able to support life and support microbes and things like that. And we also see quite a bit of change in atmospheric composition and then also carbon cycling. And the carbon cycling is particularly important because a lot of that is stemming from organic carbon burial. And so if we move into kind of the mid to late Paleozoic, so kind of the Devonian and then the Carboniferous, which is split into the Mississippian and the Pennsylvanian, you start to see these kind of reconstructions, if you're at museums and things like that, that are much more akin to kind of what people tend to picture when you kind of throw out the word deep time. You start to look at these things that kind of in your mind's eye are swamp, and often coal swamp. And that's not wrong. That's actually very accurate. But one of the things I will point out is globally, these environments weren't just coal swamps. That's kind of a colloquial term. What you're actually getting are environments that resemble our modern kind of floodplain rainforests, mangrove swamps, and then also cypress swamps. So there's quite a bit of diversity within that swamp kind of terminology, but they are a little bit more kind of what people tend to picture in these coal-forming environments and carbon-heavy carboniferous environments. And what's really interesting about these environments is they were probably existing in climates that were really similar to our modern tropical and then even our mildly temperate environments, except that they were operating under these extremely high levels of oxygen. And so it's really these environmental conditions that encouraged a lot of the lineages to grow what we kind of jokingly refer to as inexpensive or cheaply. And so what that basically means is things grew fast and they grew big. And so I have two examples of ancestral relatives down here. So the Calamites, which are related to our modern equisetum or horsetails, and then Sigillaria, which are related to modern club moss or Lycopodium relatives. But these were giant relatives. And so these giant relatives actually were constructed with a lot of air chambers and kind of their roots and their stems. And then they reproduced with what you could essentially consider abandon. So they produced a lot of spores. They produced a lot of other reproductive structures, cones and things like that. And then they just vegetatively grew just really without consideration to anything else. They put all their energy into growing really quickly. And part of that is because they had ample resources, and part of it is just evolutionarily, that's kind of how they rolled. And it's kind of this combination of this very opportunistic life habit combined with the environment at the time, which tended to be wet more than 50% of the year, that meant that these plants could grow so big, but then more importantly, die at such a rate that they were eventually preserved and buried and formed our coal. And so, like I said, this is where you get your very famous coal swamps in our modern coal as well, or modern coal. And it's not just the plants that were diversifying in kind of the mid to late Paleozoic. This is also where we see a lot of action in the fauna in the terrestrial biosphere. So this is the origin of air-breathing invertebrates. It's also when we see a rise in the amniotes, thanks to the amniotic egg. And then it's also the radiation of terrestrial arthropods at this time. And so arthropods is just a fancy word for basically insects. And so a lot of times I've mentioned this reconstruction of a coal swamp. A lot of times when you see these in museums or in talks or in lectures, you will often have just an addition of those giant dragonfly-like insects. And again, that's not wrong. Everything at this time was really just growing fast and big, which is great. And then as we move into this very end of the Paleozoic into the kind of the late Pennsylvanian and into the Permian, I want to stress that this was really a time of great transition. And so the Earth is transitioning from this deep ice house to greenhouse conditions. And it's because of these oscillating pulses of glacial and interglacial activity that are kind of in combination with what's going on during the formation of that supercontinent Pangaea that I mentioned. And this results in a very general trend from these humid kind of wetland environments of the Carboniferous, or these coal swamps, to those of increasing seasonality and in some places aridity throughout kind of the western and central portions of equatorial belts of Pangaea that kind of begins around that Pennsylvanian Permian boundary, and then also just kind of continuing into the Permian. And so one of the ways that we predict this and model this sort of climatic shift is through the use of fossil soil evidence, or what's called paleosols. And these are basically just a record of terrestrial moisture availability. And so that's how we're able to kind of have some insight into what may have been going on climatically, because it's recorded in the actual soil. And so there are a couple of studies by Tabor and Martinez 2004 and Tabor and Poulsen 2008, which are really seminal to our understanding of paleosol evidence. And in these papers, they outline a shift in paleosol type, distribution, and abundance during the late Paleozoic. And they tell us that there is a shift from highly weathered, what are called redoxomorphic pedotypes, that reflect these very subhumid or humid environments in the middle and kind of late Pennsylvanian, to soil types that are more indicative of high carbonate accumulation, which tells us they were forming under kind of a net moisture deficiency. And so I'm going to talk a lot about climate today because it's very important to us understanding what's happening on the landscape. But I want to make sure that I illustrate that the climate shift that I'm going to be talking about is in very general terms. So there's a lot of complexity when you look at any of these kind of climate systems and climate patterns. And so I want to reiterate that this isn't really a gradual change. It's more a kind of a series of fluctuations. And so they vary a lot, both spatially and temporally, in how they're changing and when they're changing. And this is likely reflecting these kind of long-term glacial cycles. So you can see this kind of general progression in the figure on the left here. And so there's a change in the paleosol type and distribution between the green and blue soils that are forming under kind of this regular or abundant rainfall, and the red and yellow kind of well-drained soils. And you can see that they kind of integrate in how they're changing through time. And we see the same thing in the figure on the right here, where we're summarizing these sort of global interpretations of paleosol data. There are fluctuations at different temporal scales, and they're kind of superimposed on these larger general shifts. So just for the sake of kind of complexity and time, I'm going to be speaking in these very general shifts when I talk about climate today. So just to make sure we're all on the same page. And what is super great about working with fossil plants is you can actually kind of consider them almost a tangible example of climate. And that's because as you know, even people working, you know, in any of the systems in any time frame, where plants grow is really a record of the conditions at that time, because they're very specialized often to, you know, different kinds of environments. And so where we find things tells us something about what was happening on that landscape at that time. And as such, the landscape of the equatorial Euramerica during the late Paleozoic is really characterized by spatial and temporal shifts in wetland and seasonally dry plant community dominance. So just as we saw with climate, there's an oscillation between these two kind of flora types that's very spatially and temporally complex. And this is really this is summarized really well in the study by Martinez et al. 2007, pictured on the right here. And it's basically, they just note that there are these oscillating swings between the different plant types that are within these different seasonally dry or humid plant community abundance. And these are relative to the climate signals. So you do see these plants are almost tracking and reflecting the climate that they're growing in. And that's really fun for us because we can go out and, you know, it's hard to dig up a weather pattern, but you can go out and you can dig up a plant and you can learn about that plant. So, okay. So where I work focuses largely in north central Texas. And so this is all early Permian strata, and was formerly a western coastal zone within equatorial Pangaea. So I've marked it kind of up in the map in the upper left there, as well as kind of in the center. And so one of the things I want to make sure I note is during the kind of early middle Permian, this region, so north central Texas, was residing at about 0 to 5 degrees north of the equator. So if you recall, it's migrated from south of the equator from kind of the Silurian Ordovician time. So now we're hovering around the equator, and then it's also tilted a little. So most of North America, instead of being kind of the orientation we know now, tilts a little bit to the side, which is interesting. And one of the reasons that we actually were able to find and locate some of the fossils that we were, because it helped kind of keep this a coastal range exterior to that inland sea that kind of comes in. And this whole area is part of what's called the eastern shelf of the Midland Basin. And the Midland Basin is just kind of the Paleozoic depositional basin for this area. And if you're from the area or if you've passed through it, you've probably noticed these very striking red beds and these very striking kind of almost like a bluish gray beds as well. And you might not have been fully aware of what you're looking at, but it was terrestrial depositional environments from the Paleozoic. And these depositional environments within the region are really variable. And so we'll actually see things, these are just two examples of two of the formations out in kind of north central Texas here. But we'll see things that vary from these kind of sand and gravel rich channel bars to environments that are heavy with point bars. And then we'll also see flood deposits and kind of floodplain deposits in this area as well. And so I want to take a look at a case study from the Emily Irish Grant locality to just kind of get an idea of how we can use fossil plants to understand this kind of bigger picture of what's happening during the Permian on the landscape. However, as billed, the Paleozoic is filled with just a lot of weirdo plants that are likely going to be unfamiliar to a lot of you. So I want to pause for a second and just take a look at some of the main groups that I'm going to mention as we go through the Emily Irish Grant locality information. And so I'll start with the true ferns. These are moisture-loving groups, and they're common kind of throughout all of Paleozoic Pangaea. And the ferns actually begin dominating the landscape way back in the Mississippian, so kind of that middle to late Paleozoic. And they radiate with kind of a huge range of growth habits. So they include things like what you would kind of almost picture in modern ferns. So you see these kind of scrambling or creeping types, these very herbaceous, smaller ones. You also see some vining things and some thicket forming ones. But then we also see these arborescent tree ferns. And so the Meridionalian tree ferns are morphologically not terribly dissimilar to modern tree ferns. So Dixonia and, oh, I'm going to forget the other one. I want to say Caetanales, but I know that's wrong. And this is the wrong audience to be wrong about that for. But anyway, so a lot of these elements, specifically the Meridionalian tree ferns, exemplify this sort of inexpensive and opportunistic growth mechanism that I mentioned earlier. So these groups really, tree ferns and some of the other fern groups, the Phyllocalians and the Sphenopterids, grow really quickly, and they tend to grow kind of with abandon and reproduce in the same sort of way. And so they're really common in the fossil record assemblages that we find throughout north central Texas and in any of the wet environments, but also kind of throughout Pangaea in a lot of the different environments, the temperate and the humid environments. The next groups that I'll talk about are the conifers, the Pteridosperms or seed ferns, and the Sphenopsids. And so the conifers date back at least to the middle Pennsylvanian, and they extend into the present. So you're very likely you're going to be familiar with conifers in some sense. They are super important and play a significant role in seasonally dry landscapes, but we actually find them globally throughout the Paleozoic and then continuing into the Mesozoic as a really important element and often an endemic element to a lot of the different floral assemblages. So they're hugely important at this time, and they continue to be important as they kind of the plant lineages evolve into modern. The Pteridosperms or seed ferns, they're a group of now entirely extinct plants, but they were pretty important during the Middle Devonian and into the early Mesozoic. And they occupied a pretty wide range of growth habits and ecological roles. And so this is another one that maybe resembles morphologically and superficially plants that we would see on our modern landscape. So the example of the Medullosin I have here actually looked likely very similar to our modern mangroves. It is not related to those, but they similarly and superficially had that same kind of look and occupied a similar niche in the environment. There were also kind of these vining and scrambling and climbing types within this family as well, or within this group as well. And so it's one that we see commonly in our wetland deposits throughout equatorial Pangaea, and then also in the tropical and temperate areas. And then finally, the sphenopsids are related to our modern horsetails. So this is the group I mentioned a little bit earlier in the talk. And they populated the landscape as both arborescent trees, so huge trees actually, but then also as scrambling or kind of these climbing vines and ground covers. And I like to think this group is kind of a survivor group, because not only does it continue into modern, so it's been alive for over 300 million years, lineage-wise, but they also have members of the group that colonized disturbed substrates. So they lived along riparian corridors or along rivers or lake edges. And they often meant that they were going to experience periodic burial or even flooding. So they were able to withstand that. But then there were other members of this group that were able to evolve and adapt to withstand periods of moisture stress. So we kind of have both ends of the spectrum within the sphenopsids. So some of them liked it hot and dry and almost arid, or at least could tolerate it. And some of them were capable of being fully underwater for a couple of days, which is quite interesting. And then the last ones I'll talk about are the Nigrathialians, Gigantopterids, and Peltosperms. And so these are ones that I'm going to bet you've probably never heard of. And that's okay, because they are so weird, and that's why they're my favorite. they're my favorite groups. And so despite being globally common in the fossil record, all three of these groups are extremely enigmatic in our understanding of their morphology, their taxonomy, and their biogeography. And so I'm not going to go into each of them too in depth, but I do want to highlight my very favorite, which are the Gigantopterids. So this is a group of broadleaf, we think, seed plants, and that grew in two kind of centers of occurrence. So there's a population where they're very common and often endemic to at Western equatorial Pangaea, or what's now modern kind of southwestern United States. And then there's another population on the other side of Pangaea across the Panthelastic Ocean that is in now modern China, or what was the Paleozoic landmass of Cathasia, where we have a whole bunch more of endemic and hugely represented fossil assemblages of the Gigantopterids. And we don't know how they're related or if they're related. We also don't know their growth habit. It's been proposed that they might be shrubs or subarborescent trees. It's also been proposed that they might be lianas or vines. So we really don't have any evidence. We only find them as disarticulated leaves. And so that's one of the things that we're hoping to answer as we keep going through and working in this fossil systems, but you never know. So, okay, so now we'll get back to the Emily Irish Grant locality and what we can learn all these different kinds of very strange looking plants. So there's comprehensive studies from the late Paleozoic floras of Western equatorial Pangaea that support the idea that Euramerica is characterized by spatial and temporal shifts between wetland and seasonally dry plant community dominance. So, for example, this study by DeMichael et al. In 2006 that I have shown here really illustrates that at nearly all times during the late Pennsylvanian and continuing into the Permian, Western Pangaea hosted plants that were adapted to seasonal drought, if not aridity. But the relationship between these kind of seasonally dry or drought-tolerant plants with wetland plants and these kind of more humid or subhumid occurring plants and their patterns of occurrence is not really understood. And so this is due largely, we think, to the variability in patterns of co-occurrence within mixed floras. And so we really don't have a ton of information on how and if these plants are growing on the same landscape, growing together on the landscape, or growing in totally different parts of the landscape. And then also how climate is impacting that. So we want to look at, so as we're kind of working in this area, we're starting to build out the picture of what is growing where throughout space and time. So it sounds like a big job, and it is a big job. So one example that we're working in is the Emily Irish Grant locality. And this is actually really ideal for looking at patterns of dominance and diversity. And it's mainly due to its uncharacteristically large size for a fossil record. So this site and this collection actually clocks in at just over 5,400 hand specimens. A hand specimen is just that. It's a chunk of rock with fossiliferous material that's roughly the size of your hand. It's a very easy metric that you can use to kind of help us create the limitations for our quadrats when we do our counts. Emily Irish was likely a small freshwater pond or an oxbow lake, which means that it was a very low energy environment. We know from the way that the fossils are preserved, or the Emily Irish collection site. And the site itself was bulk collected. And so this is actually a picture of the USGS and the Smithsonian team, and then I believe the landowner, that were out in the 1960s with their bulldozer and just basically took the entire hillside, which is great for us because it means that the collection represents a census. So there's very minimal collector bias. So they really don't, there wasn't any effort to limit the collection or cull the collection in any way relative to things that were perceived as important or unimportant or by preservation. So we have a lot of information to work with. And so we quantified the flora using what's called the quadrat method. And so we take those hand specimens, and each of those hand specimens is a quadrat, and we record the foliage taxa, or what we call a morphotype, as present absence for each quadrat. And then we just compile all those counts and basically look at what we have within the record. And so when we look at the when we look at the percent abundance of foliage summarized by clade, which I have shown here in a pie chart, you can see that ferns, true ferns, and in this case tree ferns, represent over 70% of the total abundance in the record. So it's heavily dominated by ferns. And as I mentioned, this is what we would expect. So they grow very opportunistically. They grow really close to the depositional basin, and they grow really big. So they're going to produce a lot of biomass that's going to be directly dropped and then ultimately likely buried and preserved in that fossil record. So we're not, generally, if you have tree ferns, or often a lot of the true ferns, you're going to have a lot of them if they're present. I also want to point out that there's a very clear codominance between wetland and seasonally dry flora. And so an easy way for you guys to kind of visualize this is all of the morphotypes are color-coded. So anything that kind of prefers these humid, subhumid, or otherwise wetland environments are in the blue colors, so kind of those cool colors. And anything that can tolerate or even prefers seasonality or periods of aridity are in the warm colors, so the reds, oranges, and yellows. And so you can kind of see there's a pretty even split when you take into account that kind of taphonomic bias that we infer from the tree fern domination of abundance. Additionally, I want to take a look at the morphotype bar graph, which is figure B here. And you can see that the data really it reflects what we would expect for a hollow curve distribution. And that means that there are a few morphotypes that really dominate our record. And then there's this long tail of rare or otherwise uncommon occurrences. And that's exactly what we would expect in this type of biodiversity, in this type of system. And it's what we tend to see kind of across these different floras that we look at throughout the region, and then actually throughout the time frame for most localities. And we see the same kind of distribution pattern continue when we zoom into even these smaller group levels. So there's always going to be a few species or genera that really dominate even within diverse populations. So here I have them broken out into clade, and you can just see that just visually, even without knowing what they are, you can tell that percent abundance is almost always largely based on 1, 2, or sometimes 3 things within each of these groups. And this is really significant because due to the anomalously large size of Emily Irish, we're able to get a very comprehensive look at both the species abundance and the species richness for this site. So we have a really good idea of what's going on at these sites by just doing these very simple foliage counts. One of the more unique things that we had to deal with when we were working in this collection, however, was that even though we had a census collection, so even though they took everything that was present on that hillside in that plant lens, we don't have any information about the distribution of morphotypes relative to their stratigraphic levels or their regions. So we don't know where they were in that plant lens, either stratigraphically, so by level, or throughout the plant lens. So were they on the exterior of the lens? Were they in the center? We don't have any of that information because it was all kind of bulk collected as one kind of mass, essentially. And so in an attempt to kind of determine co-occurrence, the co-occurrence structure within the record, which possibly would signal something like seasonality or fluctuations between biome dominance, depending on how these different levels potentially would break out, we plotted the occurrence of dominant morphotypes based on the abundance of these kind of wet or dry taxa per the hand specimens. And the results of this really show, and you can see it here, that the populations of wetland and seasonally dry taxa were present within the same kind of local landscape simultaneously. And an easy way to kind of infer that, even if we didn't do the math, is when we look at the fossils. So I just have some of them pictured on the right here. When we look at the actual hand specimen and the foliage as it's preserved, all of the plant organs, including the foliage, are really right on top of each other. So there's a really heavy mix within the same layers and within the same matrix of these wetland and these seasonally dry taxa. And so that's really telling us that we're finding, these things were growing simultaneously, or at least reasonably simultaneously within the same kind of local environment. So when we compile all this data and analysis, what we can do is we can start kind of hypothesizing what the landscape of this depositional environment, so Emily Irish, what it might look like. And so this is just one visual hypothesis that I've shown here, and it's based on these dominance profiles that I mentioned earlier. And then we take those and we kind of look at them with consideration to the ecological factors, so things like taphonomic and preservational bias. So in the case of the tree ferns being probably a little bit overrepresented in the fossil record relative to how many were actually present on the landscape, but then also things like growth interpretations and environmental requirements for the taxa. So in this, we look at the data we have on, cuticle or other plant structures or other physiological features that might tell us that this plant potentially was able to survive or even preferred more of a xeromorphic or what's kind of considered an arid environment. And we think about what that would mean relative to the actual depositional basin and how that's made up and where these plants would likely be growing relative to each other and relative to that actual basin. And so you end up with these kind of fun visual interpretations within each of these floras that we're looking at. And as we kind of compile them, we get kind of a better picture and these more kind of hypothesis for these region, and then hopefully just bigger and bigger regions as we're working over time. And so what do we do with this? What do we do next now that we have these reconstructions? Well, one of the things that we're doing is we're approaching the question of the relationship of different flora types with insect activity. And so to do that, we investigate the response of plant hosts to different herbivore activities through the characterization of damage type, damage abundance, and then also the host spectrum, so the different plants that are being targeted or not targeted. And we do this in an effort to kind of help us better understand the food web associations that are acting within these ecosystems. And so the synthesis that we're working on right now focuses on the frequency and nature of insect attack at Emily Irish, but then as well, we're looking at a comparison with any of the contemporaneous existing relevant floras from throughout equatorial Pangaea. And this just helps us build a better picture of kind of the environmental context for arthropod herbivory. And so the instances of herbivory when you're looking at any of these studies, are categorized into what are called functional feeding groups, or FFGs. This is largely based on the work done by Labandiera et al. 2007 and Sachat et al. 2018. And they designate these FFGs based on the basic method of tissue consumption or use. So these include things like general and specific methods of consumption of plant tissue, so things like leaf mining or skeletonizing of leaves. But they also take into consideration exploitation of plant tissue. So things like oviposition or egg laying into the tissue is also characterized. And at Emily Irish, our damage is largely confined to external foliage feeding. So I've highlighted it here, but I've also included the actual images because that's a little bit more fun to look at. And what we mean by external foliage feeding is that the consumption includes either the entirety of the leaf tissues, so in the case of whole feeding, which is marked here as HF, or skeletonizing, which is SK, or just the different surface layers, which is marked here as SF. And the insect guilds that tend to employ this type of feeding approach possess what are just kind of basic chewing mouthparts at this time in history. And they're generally just positioned outside the plant organ and consume either inward or just partially through the leaf. And so these types of damage indicate a really generalized feeding mode. So it suggests that the damage could have been done by really any array of insect groups that were living at this time. And these insects that were here had varying degrees of host plant or even tissue type specialization. So this is kind of the general type of damage that we'll see really commonly throughout the record. These aren't really the super specialized modes of attack that we're seeing at some of the other records and some of the other floras. But when we look at the actual plant host spectrum at Emily Irish, it's really apparent that the damage is heavily focused on Gigantopteridium. So that super weird one, that's one of my favorite plants ever, that looks like bunny ears, and it's marked here in red. And this is really significant because Gigantopteridium is a really minimal component at Emily Irish, but something like tree ferns, which is one of the most abundant elements in the fossil assemblage, account for disproportionately little of the total herbivory by comparison. And it's this kind of targeting of broadleaf seed plants, as we see at Emily Irish with Gigantopteridium, that we start seeing as kind of a consistent pattern throughout the region. So it's kind of this trend that we're seeing throughout Western equatorial Pangea, that's where these insects are kind of preferentially attacking broadleaf seed plants. What's interesting, though, is the plant host and the kinds of damage, so the associated damage type, really vary from site to site. So it won't always be Gigantopteridium. It'll potentially be other broadleaf things at other sites, and it won't always be this kind of general external foliage feeding like whole feeding or skeletonizing. There's all different kinds of both generalized and specialized feeding that we're finding on broadleaf seed plants and all different hosts. So there's kind of a general trend, but also we're not totally sure exactly the kind of nuances of this trend at this time. Additionally, when we start to compare kind of the regional studies of herbivory, we see that the diversity of herbivory really varies a lot in terms of type, intensity, and then feeding mechanism throughout the sites. So for example, Emily Irish, which is the largest collection that we've looked at so far, exhibits an overall really low comparative percentage of herbivory. But the Mitchell Creek Flats locality, which is a relatively small collection of fossils, and it also has a pretty low percentage of herbivory, has one of the richest arrays of damage types. So there's a lot of diversity in the insects and the feeding modes at this site, but there's not a lot of any one type of damage that we see. So we don't really fully understand what's going on there as well. And that's kind of what makes it fun, because it's these inconsistencies of herbivory across the region that might be tied to things like environmental or ecological factors. So things like depositional environment or the composition of the plant community might be impacting how and what is getting targeted, and thus what insects are being kind of drawn to that those different floral assemblages when they're living. But truthfully, we don't really know. We have these kind of broad patterns, but we haven't kind of compiled enough information yet to really have a clear understanding of what's going on with these herbivory and how these insects and these arthropod guilds are impacting these plant communities. And so one of the things we're working on to kind of address this is, well, this and other paleoecological questions, is through the NSF and the EarthCube supported Integrative Paleobotany Portal, or PBOT, which is a community-sourced and supported database that serves as a digital platform for fossil plant descriptions and their associated specimen and occurrence records. So I've included a link here at the bottom where you can learn more about this project, but essentially PBOT will serve as sort of this global database of morphological, spatial, and temporally resolved plant fossil occurrences. And that allows us for really reaching studies in paleobotany, climatology, and ecologically in a much broader way than we maybe have kind of in the past. So it's a really exciting project to be a part of. And one of the paleoecological studies that will be taking advantage of the PBOT's functionality to kind of help expand our understanding of plant resource or plant response to shifting environmental conditions is the upcoming Marie Curie action called ADAPT, or Ancient Drivers of Adaptation in Plant Traits. So this study tests the hypothesis that fire proneness is a prerequisite for the evolution of fire-adapted traits. And we do this by examining the environmental conditions surrounding the onset of branch shedding in Paleozoic conifers. So basically, this is just an effort to help us kind of understand the drivers of plant innovations and their continued function in modern ecosystem. So it's the same kind of conceptual thing that we're doing with herbivory. We want to know what's acting on these different landscape systems to help kind of motivate plant innovations, and then also kind of the onset of different trait acquisition. So what is kind of helping these plants either succeed or fail relative to changes in climate, but then also to these disturbance events. And PBOT is one of the resources that's really going to help us do that, because it's opening a door for collaboration, and it's also creating a lot more access to digitized records so that we can really work together and get a much clearer picture on what's happening in these deep time ecosystems, which I'm sure I've kind of conveyed is we don't know all that much about. We know a lot, but there's a lot to learn still. And so with that, I want to really end by reiterating that the Paleozoic is this kind of massively strange but hugely important time in Earth's history. It's where some of these major events occurred that really came to kind of dictate our future course of ecological and evolutionary history. And there's still so much to do. There's so much for us to learn, and that's what makes it so fun and exciting to work in. And so I want to make sure I give a shout out to my collaborators for part of the Emily Irish study, and then sort of some of the ongoing research that we have coming up, as well as some of our funding sources. And I'm happy to take some questions. I've also posted a QR code if you want to give me thoughts or if you have questions following the meeting. That would be great. It's just a little short platform for you to kind of communicate with me after the meeting. So.
Great work. Thank you, Rebecca. I appreciate it. So we do have some questions coming in, and let's see. So it looks like I'm going to go with this one here from the Brit Herbarium. It says, I'm fascinated by the gigantopterids, gigantopterids. I'm not sure. I know how you pronounce it, but yeah, okay. Which you mentioned, and you mentioned that they're commonly found. But it says, would you expand on why their taxonomy and biogeography remains such a mystery?
Sure. So these are one of my very favorite plants, and I actually had a whole section on them in the talk that I had to cut, which kind of broke my heart. But I realized they don't really make sense to other people because they're so weird. The reason we don't know a lot about them is we only find like loose leaves. So we've never found any of the reproductive structures in connection to any of the plant tissue. So we're kind of just assuming that they're seed plants because we find seeds in the same assemblages that we find the leaves. And we've never found any structure that really indicates, at least in North America, we don't have anything that tells us this is a tree or this is a vine. We don't have any of the kind of woody tissue that might give us that indication. And it seems like they would be too big to be a ground cover, but I mean, you never know. This was the time when the sharks had those like rolls of teeth on their face and stuff. Like nothing makes sense.
That's great. Okay, someone asks, let's see, TrimTab asks, if I find a fossil while out hiking, is there a resource you can recommend to help me determine the age of the fossil based on the location, etc.? Would that be Peabot, or is there something more like basic, like a beginner's app?
Oh, I'm sure there's an app. I don't know off the top of my head. One of the things you can do just really easily is either before you go out or even while you're out, just Google a geologic map for whatever either county or state that you're in, and that'll give you some idea at least of the age. So there's a great one just for Texas that I used. We were in the hill country this past weekend, which I'd never been, and we're driving around and I was like, what are these rocks? And so you pull up a map and you're like, okay, I'm in the Cretaceous. So I don't know anything about these rocks, kind of a feeling. But yeah, you can, one of the things that you can do when you find fossils is talk to museums. So contact us and send us pictures and send us emails, and we will try and help you whenever we can. And a lot of museums, when they're open to the public again, or as they're starting to open, they have fossil days, or they have days where the public can actually bring in things that they find and talk to experts, just usually in like the atriums and things like that. And those are really fun for us as the curators and the students and things to interact with the public like that.
Excellent. Okay, we've got another question from TrimTab, who says, I've encountered a lot of crinoids, sea urchins, and ammonite fossils in Texas. Is there a reason I've never seen a land plant fossil like you're showing us?
So if you're finding a lot of, those are all marine elements. If you're finding marine elements, you're probably just either in an older part of the section, or you're just in a part of the section that isn't preserving the fossil plants. So you're maybe not in an area where the terrestrial biosphere is getting preserved, because vertebrates, invertebrates, and plants are all preserving in very different kind of circumstances. And then there are a whole array of ways that plants and, I assume bones, I don't know a lot about the bones, but I assume bones can preserve. So they can mineralize, they can be compressed. There's a whole host of fossilization modes. So unfortunately, I don't have a good answer, except that you might just be kind of not in the right area exactly to be finding plants.
Gotcha. This is a question from me. So how does your knowledge of ancient landscapes shape how you view modern ones? Does it just change how you go out hiking and the things that how you picture landscapes?
Maybe. I assume that I'm probably pretty annoying to hike with in reality, because I'm always will go and, like, if I see, rocks or geology or any of that, any kind of sedimentological features, I will make people stop. But in fairness, because I also teach botany classes with modern botany, I make people stop and look at plants too when I find them. So I don't know that I think I'm probably generally annoying. I don't think it's specific to
annoying on multiple time scales.
Yes.
Just kidding.
Annoying throughout Earth's history.
Okay, let's see. I'm going to bounce around a bit. This one is a question from Peter Fritsch. Were any of the plant lineages mentioned affected by the Permian extinction? It was mainly marine, yes, he says.
Yes. So it was, the marine system was hit hugely at this time. And part of that is because the marine system was way more diverse. And a lot of what was going on with this end Permian mass extinction was the result of ocean acidification, we think. And so that's actually a little bit akin to what's going on now, which isn't great. But no, we lost a ton of plants. We lost a ton of insects. It's one of the biggest reorganization of plant lineages and organisms in general for the terrestrial biosphere as well. So.
We have a question about amber. So did sap or amber exist at this point to preserve any of the insects that ate the plants?
Sap and amber, I believe, did exist. So we had resin-producing plants. So we have the ancestral conifers, which are like the modern ancestor of our pine and things like that. They do find amber and things globally at this time, but I'm not, off the top of my head, I'm not thinking of anywhere that I've seen it in Texas that it's been recovered, which doesn't mean that it hasn't. It just means I can't think of it on the fly.
That's quite all right. Okay, let's see. We have a question from Joe Lippert. What's a remaining gap in paleobotanical knowledge that you want to see answered?
Oh boy. That's kind of two questions. So there are so many gaps in our knowledge, which is why it's awesome, because every time you're learning something, you learn something new. And we get to work together. It's a huge, I mean, one of the best things about working in paleobotany and paleoecology in general is it's a very community-driven field. People want to work together and we want to learn from each other because there's so much to learn and there's so much that we don't know. And there's all these different perspectives that you get to kind of experience and work through when you're answering these questions. The big one that I'm working on now is plant trait acquisition. And so that's the Mary Curie project that I mentioned that I'll be starting when I finish with the Perot Museum. So we'll be working on looking at how fire is shaping our ecosystem using some modern experimental methods and then modeling some deep time ecosystems and landscapes, just like we did with Emily Irish, but on a much bigger scale.
Great. Let's see, we have a question from Harold Keller. Are your fossils based on compression fossils from the Permian?
They are, yep. So most of what I work with is primarily compression and impression fossils. We do have a couple of sites in north central Texas that have some really beautifully preserved permineralized material that we're starting to kind of, we're actually going out to collect next week. So we'll hopefully get to have a look at some of that and some of those internal structures. But yes, most of them are compression.
Okay, here's another one from us here. Can you go back later to old dig sites and look for more of a species, or does a site get destroyed during sampling?
It kind of depends on the site. So like in the case of Emily Irish, they went in with a bulldozer and you can almost see in the pictures, there's a hill in one picture and then there's not a hill in another picture. And we've been back in the kind of the years since, and it really is, it's part of a cow pasture now. And if you didn't know where it was, you wouldn't know there was anything there. But there's a lot of regions where you'll find, especially in kind of stream channels, the way that these fossils are being buried and deposited and then ultimately preserved are in these kind of almost like little pockets. So you'll go and you can maybe find a plant lens of a couple hundred or even a fewer hand specimens in one spot, and you'll collect it and bring it back. But you'll go out again in the future in the same kind of just very local area and you might find another plant lens. And so you kind of never know what's going to preserve and what's under the ground until you start digging.
It reminds me of when you grow potatoes that you're like, well, I don't know if they're ready until I dig them up, and you kind of destroy something in the process. But you know, you gotta get it. You gotta get it somehow. Here's another question: how do you find new dig sites? Private landowners, do they find stuff and contact you, or do you ever use a modeling approach to landowners to tell them to keep an eye for things and call you if they find them?
That's a really good question.
Did that make sense? Yeah, sorry.
Yeah, no, I love that question. So there are a lot of ways that we actually find dig sites. So one of the ways is we have an idea of where we would expect to find fossils based on, you know, these people that are, you know, we have all these experts that are way smarter than I am on the geology and the sedimentology. So they can look at the landscape and tell you, okay, this used to be a channel and it's filled in now. And we can tell because of X, Y, and Z or whatever. And so we know likely in that channel, if there are going to be fossils, they'll likely be in this area. And in a broader sense, we have some idea of if it was a marine system or a terrestrial system that we're working in. And so, you know, especially in the southwestern United States, there's these depositional basins where we know the broad kind of parameters of where we might find Permian fossils or Paleozoic fossils or other time frames. And we do have on kind of a more local scale, we do have landowners that will contact us when they find stuff. In fact, we just had someone at the Perot Museum that found some really cool fossilized wood material, and they volunteered it and brought it in and showed it to us, and we're going to go out and check out what else they have out there. And so it's really helpful because there's, you know, obviously in Texas, there's a ton of private land. And so there's a lot of stuff that we can explore without the help or the cooperation of the landowners. And we love working with, you know, we have some of the most incredible amateur collectors I've ever worked with or ever experienced. They're so smart and so wonderful and helpful. So yeah, there's a lot of ways we find sites. And sometimes you just find things because someone was, you know, building and needed to dig a hole, or their kid was out playing and they dig a hole. You never know.
All right, we've got a last question here. Says, that was really great. Thank you. I appreciate the imagery and orientation. Do you and the Perot offer field trips or guided hikes for the paleo novice?
I think pandemic-wise right now, I know we're definitely not doing that, but I also know that they do take a lot of volunteers. So the paleobotany program is relatively new. It's run by Dory Contreras, who I think is on this call, and may chime in here with some more information about this. But I know they have a ton of vertebrate and invertebrate volunteers that work with them, and that's kind of, they get to go out on digs and they get to come out and kind of either work with our existing collections or go out and kind of prospect some new things and help recover things when we find it. And I believe in the future, the steps are to kind of involve some of the paleobotany community as well.
Great. Excellent. Let me give a quick check to see if we skipped anything. I wish I could mark these as being done. I believe if there's anybody who didn't get their question answered, you can repeat it in the chat here. You can type it in again. But it looks like we've got most of them. Check really quick. No, I think that's it. Hooray. Oh, and look at that, just right on the dot, 100. Well, that was super fantastic. I mean, you've done amazing work. That was very intense, and there's a lot going on there, and I can see how there's so much out there, so much to explore. So how do you staying focused, right? Is probably something that you struggle with, because there's, and you have all these collaborators who are asking their own questions. You're like, oh, okay, well, I can look at adaptations, and I can also look at, but there are all these things we learned today that are just about like the plant, the insect damage, the leaf holes, and something that strikes me every time I engage with a paleobotanist is, how do you struggle with we don't necessarily know that it doesn't exist. We just don't have any proof of it yet, or it didn't, that act or that behavior or that species or whatever, just didn't fossilize, that's a real job hindrance.
Yeah, I mean, it's kind of a double-edged sword. It's a very cool thing, because at any time you could walk out and find a fossil that changes everything that you know about something. But all of those are underground. So everything that you're working with is underground, theoretically. So.
Right. Well, is there anything else you'd like to promote? You got any events coming up at the Perot there that?
Nothing that I'm in charge of.
You just finished a big conference. Yeah. I'm aware. That's great.
Yes, yeah. So we're getting a little bit of a break to get to go out in the field and actually hike around and dig up some rocks, which will be really fun. But I do want to thank you for having me. I really enjoyed this.
Absolutely. We're so thankful to have you here and Dory, and to have, our colleagues, making better connections with our colleagues at the Perot, just right down the road, right? We hope you'll come and visit us in person sometime when it's safe to do so.
Yeah, absolutely.
All right. Well, we'll say goodbye to the audience now. Thank you all for joining us. Don't forget to join us again next month. I was going to say week. Next month on June 1st, when we'll do this again. All right. Thanks, everybody. Bye-bye.